It turns out that at Sierra College, Ray Underhill, my predecessor in the biology department, started a student research project, and my wife was one of the two first students participating in the research project. One student was working on the life cycle of intestinal parasites in porcupines, and my wife was trying to make sense of all those funny looking growths that were showing up on the oak trees around her place. And it went from her research project at Sierra College to my master's thesis to me putting in one job application after I got both my degrees at Davis. And I got hired and spent 50 years there on the faculty, usually about as happy as a pig in a mud hole. Yeah. How do we advance?
Oh,
Which is the advance button?
There you go. Well, what's going on? Okay.
Oh,
You know what? Just click on here.
okay.
Okay.
Sound locally. It turns out there are a number of things that can stimulate plants to grow. And going on to the Davis campus, there is one of the agrobacterium tumors on a big tree that is so big I couldn't reach around it. And there's an example over here on the countertop that came from a tree at the fire station the other day. It turns out this willow pine cone gall is caused by a midge. And it turns out that what happens is the leaves go ahead and develop in the bud, but the stems don't elongate. And so you have this whole cluster of leaves with no stems in between them. Insects are able to produce a number of chemicals that can stimulate plants. Sometimes we notice them, sometimes they sort of go unnoticed. Aphids form galls on cottonwood leaf petioles. And so about this time of year, as the cottonwood leaves are falling on the ground, if you see those big swollen petioles right at the base of the blade, if you open it up, you'll see aphids inside there. It turns out typically the insects that have incomplete metamorphosis produce open galls. So there is some place to crawl in and crawl out without having to chew through any plant tissue. The ones on the bottom are manzanita, and if you're careful, you can sort of peel it back and see the aphids in there. Set it up for the sharing here. Are we broadcasting to the members that can't come tonight? Yes. Yeah. Okay. Gall you find might be formed by a mite. And typically mite galls are open on the bottom, project on the upper side of the petiole. They don't have chewy mouth parts, so they need an easy exit for getting out of their homes. Whoops, I went one too fast. There are gall wasps that attack rose bushes. I have never seen them on the standard commercial varieties of roses. But here in California, the wild roses fairly commonly have them on them. And there are a number of species. Diplolepis is the genus for them. Bicolor is the species name. And you can sort of tell why it was named bicolor. thorax is black and the abdomen is red. Beautiful little critters. A lot of times when people hear the word wasp, they get all paranoid because wasps and bees and ants can sting. But in the case of these gall makers, the stinger is really an ovipositor. It's just an egg laying thing. So none of them are going to give you any problems. Most of the cynipid wasps produce galls on oak. You can find them on the petioles and the leaves, on the buds, stems, roots, acorns, male flowers. Most anywhere you look, if you look long enough, you can find them. There are at least a couple of hundred kinds, fairly well known here in California, and there are a number that have not yet been described or named. I still have 5 or 6 names in my thesis that haven't been used yet because they haven't been published. And in a while, I'll show you why. The galls are a way for the insect to provide their offspring with a stable, secure place to live, food supply, and in some cases, protection from predators and parasites. And the plant can grow this material off somewhere away from the main growing parts of the plant to minimize damage to the plant. There are a few species that attack chestnuts, and those of you that know Castanopsis, the other member of the oak family here in the state, there are some nipples on both species of Castanopsis. They are not very well known either. There aren't many folks take the time and energy to study these things. So if anybody is interested, there are still plenty of opportunities to actually publish some papers on this. Anthraquinus is probably the best known one in the whole state. You can't live in California very long and not notice those things. We sometimes call oak apples. The illustration on the screen here for the insect is way too big. And there are a few insects emerging over here in some of the containers. They typically start emerging right around Halloween and will keep emerging through most of the month of November. Each particular species, it's got its own distinctive pattern when they come out. Neuroterus saltatorius is a tiny little thing. You've probably heard of Mexican jumping beans. Well, it turns out that the saltation is jumping. Neuroterus saltatorius falls on the leaves in the summertime. And the larvae inside don't like sitting there in the heat of the sun. So they curl up and straighten out and curl up and straighten out, and they will jump their gall off into some more hospitable place for them. So it's interesting to see the curve just jumping with these things. Up in El Dorado County, some of the beekeepers up there found wasps, bees, paying a great deal of attention to those galls in the bottom right-hand corner. It turns out that those galls secrete honeydew in the fall. It's a sugary solution. Ants love it. Ants will show up. Bees and wasps will show up. And what that does is that discourages the parasites from coming along and laying eggs down inside the gall to do in the gall maker. And some of the beekeepers, when they would find a tree that had lots of that, would actually take their beehives over, put next to that, and let the insects collect the honeydew from it. It turns out that it is not sellable as honey because it isn't flower nectar that's producing it, but it's enough to help get bees through the winter if need be. Surprisingly enough, the life cycle of Andricus californicus, this big one on the left, as far as I know, is still not known. Nobody seems to have taken the time to collect the insects, put them back on the tree, watch for sure where the female oviposits be what comes out. Because not all of these things breed true each generation. Uh, there's an interesting pattern with these things. This little tan colored gall with little prickles on the sides of it is on the interior live oak and the coast live oak. And some people have been known to collect these things, paint them up, decorate their Christmas trees with them as Christmas ornaments. And then they wonder why it is that between Christmas and New Year's, there's holes in their Christmas decorations. And there's all these funny little insects on the windows trying to get out of the house. Well, it turns out when those females emerge, and you only get females out of there, they're black. They emerge about Christmas time, and they will go find leaf buds and oviposit on leaf buds. And when the leaves are in the bud stage, it's the outer surface of the little leaflets that are accessible. And so the females lay their eggs on the underside of the little leaflets in the buds. And later on in the spring, if you walk around and look in the live oaks on the underside of the leaves and look up, there's little sort of toadstool things with a red rim on them, and the rest of the gall is sort of a greenish color. What comes out of those in the springtime is males and females. They do not look like their mother. They're anatomically different. They're different colors. Um, it turns out that these females produce eggs by meiosis. The eggs only have half the chromosomes that they had, and so their offspring are haploid, kind of like male honeybees are haploid. And so the spring bisexual generation have produced gametes by mitosis. They mate, the females lay diploid eggs back in the twins that have developed after the buds opened up. And so you alternate from a unisexual generation to a bisexual generation back to a unisexual generation. The reason the ones that are in my thesis that only have manuscript names is I was finding things that nobody had ever seen before, but I wasn't taking enough time to put them back on the tree to see what came out as the next generation. The unisexual generations are quite conspicuous, fairly well known. And any editor worth his salt in an entomological journal, if I sent in a paper with a half a dozen names of new bisexual generations, he would say, those are probably already named for the unisexual generation. Why don't you go connect them up and then we'll publish your paper? I haven't gotten around to it yet. So unisexual generation females, fairly good size, robust, big fat abdomens, lots of eggs. Meiosis to produce haploid egg produces bisexual male female generations. A lot smaller. Relatively smaller abdomens also. And this is the norm for most of the life cycles that have been verified. There are some that do not have alternating generations. The ones on the rose bushes are just bisexual all the time. A few of them have been verified to be the unisexual generation all the time. And there's a whole lot that still haven't been verified either way. Andricus kii typically occurs on the underside of our valley oak leaves. And it's these kind of cute little volcano conical shape, sometimes sort of cream colored, usually sort of red. And once in a while, it's on the pink side. And what comes out of these are all females. Here's one that used to be called Andricus kingi, but it's on the blue oak. And it looked enough like the ones on the valley oaks. People said there's probably just a little bit of variation because of the host differences. Well, I got around to rearing out the insects and looking at them. And the two insects that came out of these two different kinds of conical galls were not the same thing. And one of the trees near the train tracks down in Davis, isolated all by itself. There wasn't another oak tree within a mile or two. It was just loaded with these things. It was the only kind of galls I could find on the whole tree. And so I said, that's nice. There's not much competition. When I go back and look at it this spring, I'm pretty sure that I'm going to find the bisexual generation of this thing. And lo and behold, that next spring, that tree was just loaded with these little delicate uh, spherical things on the margins of the leaves. And if you cut them open, inside was a blue-black larval cell suspended on little fibers. And from that, I managed to rear out the bisexual generation. So Andricus kingi was the name for the one on the valley oak. It turned out this thing had not been given a name. The bisexual generation was already known, but when you got the females, they belonged in the genus Andricus. And so I got to modify the taxonomic placement from this. So it became Andricus atramentus. And not too long ago, the Davis Library sent me a letter and said, we would like to digitize your thesis. And they wanted an electronic signature on the letter they sent me. I'm still old school. So one of these days, I'm going to sign the paper and mail it back to them instead of doing the electronic format. Um, and I don't know if it will encourage anybody to go continue working on this, but there are 5 or 6 manuscript names that are 5 or 6 more papers waiting to be published someday. If somebody will just do the next step. And I would be happy to work with them if anybody wanted to tackle it. Okay, I already told you that one. So there's a reason I brought all of those gulls down here tonight. One, they just keep cluttering up my lawn. And so I'm cleaning the yard with it. But since they are emerging now, if you happen to have a valley oak or a blue oak somewhere in your backyard, you can take up some of those home, rear out the insects, go put them out on the tree, watch and see where they oviposit. If you have some of your old nylon stockings, you can slide it down over the branch with the insect in it so they can't get away, and to keep other things from coming in to lay eggs inadvertently. Uh, how many of you have a greenhouse? One greenhouse. If you had a little pot of oak tree at home, you could put it in your greenhouse and you could work on that in your greenhouse instead of being out in the yeah. Um, I don't remember if it was Caesar that said all Gaul is divided into 3 parts, but it turns out that the oaks in California are divided into 3 subgenera. The white oaks have got fairly light colored rough bark and fairly light wood, and it doesn't make as good a firewood as the black oaks. And the black oaks have a darker smoother bark. Sometimes it's red colored wood, which is why some of them are called the red oaks. And then the intermediate oaks are only known from Western North America. Quercus chrysolipus, um, Quercus vaccinifolia, huckleberry oak up in the Sierras. If you get down into the desert, Quercus dunnii is a desert oak. And interestingly enough, there is an oddball grove of Quercus dunnii over at Wilbur Springs, north of Clear Lake. And when I found those things, it was sort of like finding a saguaro cactus north of Clear Lake. They are a desert oak. And for those of you who ever get down below the border, Orchis cedrosensis is a Mexican oak that has a number of insects on it that are not adequately known yet either. Now, the taxonomists did this all by themselves, just looking at the oaks. The wasps are also plant taxonomists. Something that occurs on one of the white oaks will quite often occur on many of the other white oaks, but never on any of the black oaks or the intermediate oaks. Those on the intermediate oaks can occur on a number of species in that group, but never on the other two. Same for the black oaks. Typically can occur on a number of species in that subgenus, but never on the others. And as I went through the literature once upon a time, I found one listing of one species that occurred on two subgenera. And I said, that's really weird. So I got hold of the insects from both trees, and lo and behold, they were not the same thing. They were misidentified. So one name was valid, the other one wasn't. And so that was another one of those papers. And the wasps are really good chemical plant taxonomists. So here's the collection that we've got here in California. Sadler's Oak is up on the north coast, Trinity House unit area. And so when I got up to the ranch, I was going to plant all of California's oaks at my place so I would have my own rearing station. And then I got sidetracked with teaching at Sierra and working at the museum and 5 kids and 38 acres to keep track of, and a number of things didn't happen. Okay. Over on the sidewall are a number of Riker mounts that have examples of mostly cynipid galls, but there's a few of them that are not produced by the cynipids. Those need to go home with me. But the galls in the buckets and the boxes, you can take with you if you'd like. Whoops. How do I back up?
Which one did you need to get to?
There we go.
Okay.
Ron Russo used to work for the East Bay Parks and Recreation District, and he loved photography. He loved the outdoors, and he was quite fascinated with plant galls. So he put together the field guide to plant galls of California and Western states. And the University of California published a version of it. And currently, can't remember which university it is. Anyway, there's a copy of Ron Russo's bigger book over here on the sidewall. So if any of you are interested in it, Russo has 2 volumes out on plant galls. Great color photographs, names of the things, a little bit of natural history, tells you what the host plants are. And they're just interesting, curious, and it would be really nice to know what chemicals it is that's modifying the growth of the plants. Because have you ever heard of cancer? This is essentially a plant version of cancer. These things would never have been produced if it weren't for the insect stimulus. And so um, it's turning out that they're either RNA, and in some cases DNA, is helping in this process. Um, they're probably making small mobile transfer RNAs that are messenger RNAs that are being um, able to be decoded by the plants. Now, what do you know about cuckoos? Cuckoos lay their eggs in somebody else's nest. And what happens when the little cuckoo bird hatches out? The other birds snuggle up against it, and the cuckoos have an interesting stimulus. You push on their back, and they push back. So the nest mates snuggle up against the baby cuckoo. The baby cuckoo snuggles up against and keeps snuggling until the other baby goes over the edge of the nest, and then they go back and settle down. And so it turns out there are a number of cynipid wasps that cannot induce their own gall formation. They find somebody else's gall, lay their egg in there. Sometimes both of them can develop. Most of the time, the inquilin takes over, crowds out, eliminates the original maker. And so you can collect some of these things and you get out an insect that when you key it out, it says, this is not the gall maker. There's one of those on rose bushes. It has a conical woody gall on rose bushes that you never get gall makers out of. So I started trying to figure out what was happening and managed to find a little aborted bud gall that was yielding the gall makers. And if I could keep them from being attacked by the inquilins, we got another species of gall wasp for the rose bushes. Now, after your gall makers come out of the California oak apples, you might be tempted to throw them away. But typically in these galls, there's a whole community of parasites that have come along, not inquilins to live with, but parasites to actually parasitize and kill the things. And that Pterimus female down there in the bottom right has got an ovipositor that's longer than she is. When she finds a gall, she can reach down through there and deposit an egg on the larva down inside there. Her egg hatches, consumes the original gall maker. And so sometimes a month or 2 or 3 or 6 months later, out of these galls, you'll start getting the crop of parasites. Do any of you like walnuts, pecans, or almonds? Yes. Do you have a walnut, pecan, or almond tree at home? It turns out that the commercial nut growers do not like Andricus californicus because it is also a host to the filbert moth. And so when they have an orchard of nuts surrounded by valley oaks with all of these on, just about the time the hulls begin to pop open on their nuts, the filbert moths hatch out of here and go attack their nut crop. And if they don't get them harvested, you get the filbert moths growing. So there are all kinds of things going on in those plant communities. These parasites have some really impressive colors to them. Some are metallic green, metallic blue. Some of them have a blue thorax in a metallic purple abdomen. The cynipid female's ovipositor is long enough to get an egg down into the plant tissue. The parasites need to have ovipositors to get down through however size the gall has gotten to be so they can deposit their eggs in there. So it's kind of fun. And while I was in graduate school, I was rearing out the insects, the gall makers, but I was getting lots and lots of parasites. Another grad student across the aisle from me was working on parasites. And so he was raising gall wasps to get the parasites. So he gave me his gall makers. I gave him my parasites. I named one after him. He named one after me. It's considered poor frame or poor form to name them after yourself, but we named one after each other. And so both of us are in the literature. And that concludes the slides. But over here on the sidewall, we have some things that I would love to show you. And I can't very well stand here and show you those things. So maybe we decide the formal program is over and we just go an informal thing on the aisle.
I think so. And you know what?
Or I bring them over here. And,
that could work too. I can.
are we about out of time anyway?
No, we still have a good 5-10 minutes. Let me see.
Stop screen.
Oh, stop screen share. Okay, thank you.
Any questions you folks have got?
So on the graphic was you showed the relation of some of the life was the triangle was so that is one some of the water the base rather than being in a single phone. You know why that is when you become out of this? Yeah. I think you can't help me up to the it's not because they're diagnosing diseases and when you got the general store, they're very taking the same ones. Very like us.
The last question is more about it. So for that same grouping, they also made the little marginal round stalls that are other like cycle have.
So the sure and ready see, I still don't know. I still don't know my second and rather than horrendous, we know it's like a moon, but I don't know something for the others. And it turns out, you rather than likely, association of California's name once to the insect that live on those things.
Charles, do you have maybe I can bring some just for a couple for our Zoom.
You finish?
I
In here are galls that I have collected from my driveway. This is about somewhere between a third and a quarter of the crop from this tree this fall. They started dropping about the 1st of October and have still been falling. And the chickens have eaten some of them that I didn't get to. And the car ran over a bunch of them that I didn't get to. But I even brought some empty fill bites. And if any of you were interested in seeing what these things look like, oh my. Now you can do what I'm
getting everywhere.
on the carport. And most every trip across the carport would get me another teaspoonful or another tablespoonful. And if you keep these things at home, all closed up like this, the moisture will come out, it'll get moist inside, they will get moldy. So you don't want them in a totally airtight, waterproof container. And sometime during the winter, they will emerge in the garage about the same time that the buds are starting to pop on the trees, because the alternate generation of these is in the female flowers. No, I'm sorry, on the male flowers. Now,
Is this is there a specific tree it's associated with?
This is off the California, or the interior live oak with the
Interior. Okay.
And
Do you have to have an interior live oak?
if you had the Quercus agrifolia, the coast live oak, it will work on that also. It occur on both of them. Now, what was Clark Kent noted for? Being Superman, and Superman had X-ray vision. If you would like to have X-ray vision to find out what's going in here, you go to your local pharmacy, buy a package of clear gelatin capsules. Carefully slice this open, put the larva in the gelatin capsule, get a roll of double stick tape and tape it onto a 3x5 card, something like that, and put the gelatin capsules on the sticky tape. And one of the first things that you'll see when they start to pupate is the pigment in the eyes develop. And you'll see these two red spots on the anterior end of the things. And then they will pupate. And when you see them transformed into adults and moving around inside the gelatin capsule, you can say, oh, the ones that are still inside here are probably about for that same point in time. And then you can check to see if you've got your little potted oak in your greenhouse, so you can put those things on the tree. Now, if your little potted tree is not mature enough to be producing male and female flower yet, that may be a problem of being able to work on the life cycle.
So do these damage?
Well, each one of these wiped out one bud because there used to be a bud where the egg got deposited. But it turns out that many buds on the tree has not done the tree in. It's still doing fine. Some of them will cause a little bit of damage, but in most cases, the plant has just managed to move these things out, put some tissue out there to minimize the damage to the tree, and they typically get by quite nicely.
I'm sorry. Did you finish answering that question? Did you finish answering that question?
I think so.
Oh, okay. There's a question online. It says, can you speak to the extent to which the presence of galls indicates overall ecosystem health, specific host health, and also at what point would gall cover indicate the host is doing poorly?
I have a valley oak that is so heavily infested with the Andricus californicus, the big oak apple galls, that I think it probably is being somewhat of a detriment to the tree. It also is right next to my lawn, and the lawn gets a little bit of water. And as we know, watering our native oaks that are used to dry, hot summers is not a good thing for them. And so I'm not sure how much of the problem is a problem from the galls and how much of it from the fact that it's growing beside my lawn. But it's a wonderful crop of these things. And so the tree has been there for 56 years since we moved into the place, and it hasn't died from it yet. So I don't know of a case where the galls have killed any of these trees. I don't think there's a serious detriment in most cases.
So this year I noticed on some valley oaks over by the river, were so covered with the red cone gall wasp that they were pink from a distance. And I was like, why are the valley oaks pink?
Yes.
When I got close, they were just furred with red cone galls. So I was wondering, just like oaks will mast.
Is that this year or
This is
previous
this
year?
year. This is this year. This
Oh
fall.
my, that
So-
would be a really good tree to go work on figuring out what the rest of the life cycle is. So the real Andricus kingi.
Unfortunately, it's on a bit slow, so it's hard to get to. But do they sometimes mast the way that acorns matched on groups of oaks?
There are some really interesting examples that relate to acorns. Again, up in El Dorado County, some of the folks noticed that acorns are oftentimes lopsided. And when they started looking at these lopsided acorns, pulled the cap off, just underneath the cap, there was a larval cell where an insect had been developing. It had drained off enough energy that it had kept the acorn from growing straight. It had bent over. And I managed to figure out what the life cycle was on that one. Interior live oaks take 2 years to mature acorns. Most live oaks will do it in a year. And 1 year we had a massive acorn crop, and there were lots and lots and lots of them that were all lumpy. So as I cut them open, it turns out that the acorn inside had basically lignified, and it was just filled with larval cells from some sort of a cynipid wasp. Turns out it's Caloritis milleri. And I collected a whole bunch of these and wanted to rear them out and find out what the next generation was. But that next year, nothing came out. I thought, darn, did they all die? I didn't think I'd been that hard on them. They live out there in the environment through all kinds of tough times. And so I didn't throw them away. I had too many other things going on. The second year, I got a nice big emergence from all these acorns. Took them 2 years to finish their development and emerge. And so I put some back on the tree and figured out what the life cycle was. Didn't throw the acorns away. Year 3, nothing happened. Guess what happened in year 4? I got another emergence from the same batch of acorns. Guess what happened in year 5? Nothing. Guess what happened in year 6? I got a few more. So it turns out there's all kinds of neat adaptations. These wasps are tuned into the fact that the tree takes 2 years to make acorns. You wait a couple of years. It's kind of like when you want to go out and collect pine nuts. It takes 2 years to mature the pine cones. So if it was a really good year this last year, you probably want to wait 2 years for getting the next real crop. There's almost no part of the plant that is immune. There are some that are on the roots. Roots are not very often exposed, but when they are, there are some that will live there. We had a couple of trees that had to be taken down for PG&E's reasons, and the stump sprouted all kinds of growth on it, all vegetative growth. It was not doing any reproduction, male or female flowers. And I found a whole bunch of stems that had been attacked by insects. And now that we've had a good rain on the interior live oak, some of those stems over there that are laying on the countertop have got ragged splits in them. Now, there's a wasp that goes lays its eggs in the stems, and about the second good rain, the tree soaks up lots and lots of water. The turgor pressure inside the plant increases, and there is a little hard brown larval cell. A little hard brown larval cell that gets pushed right out through the bark, flops out on the ground, and then sometime later on during the winter, the insects will chew their way out of the larval cell that fell on the ground, and nobody knows what the next generation is. So there are others that instead of making big ragged stuff like this, all you'll see is a tiny little hole. I was at my parents' place one spring and looking at some of the newly grown branches that were up right by the edge of the railing on the deck. And there were little wasps flying around. I could recognize them as cynipids. I saw holes in the stems where they'd been chewing their way out. I said, oh, I know nobody knows who this is. And so I got my little aspirator jar, sucked up 126 of them. I didn't have time to sit and wait for them to lay their eggs and do all that sort of thing. But I said, I want to at least know what the insects are. Took them home, mounted them up, looked at them under the stereoscope. 126 males. The males emerged first. I said, I got to get back up to mom and dad's to wait when the females come out and collect them. Didn't get back up there in time. I have 126 males of an undescribed bisexual generation. Still have not found the rest of it. There's plenty to keep busy with. I was thinking that when I retired, I would get back to doing some of this. But now that I'm sort of running the ranch by myself, and last year we had 29 oak trees fall over. This last winter we had 9 oak trees fall over. I have not had much time working on gall wasps. I've mostly been making firewood. So anyway, there's plenty yet to do. And if you would like to take any of those things home with you, rear a few out. And if you'd like to work on a life cycle, I'd love to work with you. Thanks.
Awesome. And thank you everybody for joining us on the live Zoom.
The recording.
Ah, thank you, because I forgot. Good night, everybody. Stop recording.